Very good afternoon, dear students. We have been discussing about unit number 3 of Microbes and Environment paper, the open course for you. And in unit number 3, we have discussed about the importance of water and the quality analysis and standards of water. And we have discussed what fecal coliforms and waterborne diseases in the previous classes. The next important topic mentioned in the syllabus. This is very important and interesting, and this is the one of the routine activity of the mineral water companies as well as the testing of water, especially the drinking water. Okay, that is indicator microorganisms or indicator organisms and sanitary examination of waters. So how will you test the quality of water, whether it is safe to drink or not? And this is known as the potability testing. Potability means the water potable means the water which are very safe to drink. They don't have a contamination, especially the bacterial contamination, and that bacterial contamination is in a permissible limit that not exceeded by the World Health Organization's level that is present in the water. And that is the next important topic whatever we are going to discuss. And before we are going to start, we should know what are indicator microorganisms or indicator organism. As the name indicate, indicator, which indicates something. What is that something? So that is what we are going to understand. Okay. So indicator organisms are microorganisms whose presence in water indicate the probable presence of pathogens or disease causing organism. In the previous class we have discussed, if you drink contaminated water, you will be having so many diseases, okay, especially bacterial diseases. So many bacterial diseases, waterborne diseases. So there are certain organisms known as indicator organism. You detect those indicator organism that will tell you whether the water, whatever we are going to use is going to have the pathogen or disease causing organism, and that will indicate the presence of pathogen or disease causing organism, and such organisms are known as indicator organism. Then this indicator organism should be, they are non-pathogenic, it should not cause any diseases, the organism should not cause any diseases, and which occur consistently in pathogen contaminated water. Wherever pathogen, bacterial pathogens are present in the water, this indicator also should present, and they should be in a position to survive in such kind of water. Okay, and it should not multiply in water. Okay, so it should have, but it should not multiply, and which are reliably detectable even at a low concentration. Even if the concentration is very low, it should be possible to detect. These indicators should be detected and are present in greater numbers than the other organism, and it should have a more number for the easy detection in that particular water, and which should have a similar survival time to the pathogen. Okay, and how the pathogen is surviving in the water, this indicator organism also should have the similar surviving time and surviving density. And mainly this indicator bacteria are known as fecal coliform. This is what we have discussed in the previous class. So fecal coliform group has been used as an indicator of bacterial pathogen presence and general wastewater contamination. Okay, so the presence of bacterial pathogen can be detected by checking the presence of fecal coliform count. Okay, if you see the fecal coliform in water, that indicate that that water is having so many types of pathogenic bacteria. So that are indicator microorganisms. And what are the criteria for considering this bacteria or indicator microorganism? What are the major criteria in which we can consider for an indicator bacteria? So what are the criteria should fulfill if we wanted to call a bacteria as indicator organism? There are 9 criteria. More than that, some of the important criteria we can discuss. Number 1, the indicator bacterium should be suitable for the analysis of all types of water. We are going to check so many types of water. This indicator should be suitable for the analysis of all type of water. What do you mean by all type of water? That water can be simply tap water or pond water or river water or well water, ground water, recreational water or sea water or waste water, etc. Okay, so any type of water, the indicator organism should be suitable. Next one, the indicator bacterium should be present wherever the enteric pathogens are present. Wherever there is a pathogens are present, the indicator bacteria also should survive there. Then only they can detect. Along with the pathogen, they should be present. Next one, the third point, the indicator bacterium should survive longer than the enteric pathogen. Enteric pathogen means, you know, that the bacteria that causes gastroenteritis, okay, loose motion, diarrhea, okay, dehydration. As soon as you take the water, you will be having all these problems. We have seen that. So this indicator, if you wanted to call certain bacteria as indicator organism, it should survive longer than the enteric pathogen. In comparison with the pathogen, the indicator bacteria should survive more time. Next one, number 4, the indicator bacterium should not reproduce in the contaminated water and produce an inflated value. Okay, indicator microorganism should not reproduce in the contaminated water. In contaminated water, it should not reproduce and should not give a false positive result. Then the assay procedure for the indicator should have a greater specificity. Other bacteria should not give a positive result. Okay, this is very, very important. The method for a test, that is known as assay. What methodology we are going to test this particular bacteria, that procedure. Okay, when you are testing the bacteria using that procedure, the test should be very specific. Okay, the test should have greater specificity. Other bacteria should not give a positive result. Okay, so only indicator microorganism should give, certain bacteria should give the result, other bacteria should not give. You know that water consisting of so many bacteria, not all organisms are known as indicator bacteria. Okay, only a small group of organisms are known as indicators. So that is what I told. The assay should be very specific and it should not give a false positive result. Okay, then the procedure should have high sensitivity and detect low level of the indicator. Even if the low level of indicator, it should have it should be detectable. The methodology should have high sensitivity. Okay, even if you have a very low number, it should be possible to detect. Next one, the testing method should be easy to perform. This is very, very important. Okay, the test, a common biology person, even you people also can perform. Don't think only microbiology person should be performed. Even any biology person should be able to perform. The testing method should be easy to perform. It should be in a very easy to perform. Next one, as I told you, it is non-pathogenic. The indicator should be harmless to human. Okay, the indicator bacteria should be harmless to human. It should not cause harm. It should not create a pathogenicity. It should be non-pathogenic. Okay, the level of indicator bacterium in contaminated water should have some direct relationship to the degree of pollution, especially fecal pollution. Okay, so if you look the level of indicator, concentration of indicator microorganism per 100 ml of water, that should have a direct relationship with the pollution, especially bacterial pollution present in the water. So those bacteria that has all these features, such kind of bacteria are known as the indicator bacteria. Okay, such kind of bacteria are known as indicator bacteria. And this we have already seen in the last class. What are these indicators? That are known as coliform. What are coliform? Coliforms are bacteria that are always present in the digestive tract of animals, including human, and are found in the water. Okay, I told you coliforms are gram negative non-sporulating facultative anaerobic organism. Okay, they should be present in the digestive tract of animals, including human and warm-blooded animal. We have seen this in the previous class. Okay, here you can see one class of this one, and you can see lot of pathogenic bacteria. If you grow in a microbiology laboratory, you will be able to see lot of organisms, such as bacteria you can see, fungi you can see, all those things you can see. Okay, and sometimes that is indicate total coliform, fecal coliform, and E. coli. Okay, the most basic test for the bacterial contamination of the water supply is a test for total coliform. That is what I told you. Okay, so in order to check the water is a good quality water, just what you need to do, just you count the total coliform bacteria present in the sample. This total coliform count give a general indication of the sanitary condition of the water. Okay, so how will you get to know that the water is safe to drink? Okay, water is a good quality water. Okay, so if you check the total coliform count, if you check, you will be able to tell the water is good to drink. Okay, this is very important, because every day in a 1 hour, at least 1 hour, how much water we are drinking, imagine. So that is what this is very important. And without this particular test, none of the water processing companies can survive. There is a microbiology testings are involved there. Now we are going to see how this water is examined in the laboratory. Okay, how big company they are preparing the water and how they are examining the water. What is the sanitary examination of water? Okay, what are the steps that is involving in the sanitary examination of water? Okay, that is the next topic mentioned in the syllabus. So what is the sanitary examination? That is the bacteriological water analysis. The presence of bacteria that is commonly present in the water can be detected. Okay, the bacteriological water analysis, it's a method for analyzing the water to estimate the number of bacteria present and find out what sorts of bacteria they are in the water. Okay, so that is the help to analyze the water to estimate the number of bacteria. 100 ml of water, how many bacteria are present? What is the number? Okay, that is known as total count. Okay, so and what this bacteria are, what sorts of bacteria, what is the bacterial density in that particular water. This clearly represents the water quality standard. Okay, so this is the way we can tell the quality of the water. Okay, water quality standard. And it is a pure microbiological analytical procedure which uses samples of water, and from the samples determine the concentration of the bacteria. Any sample you can collect. Okay, in your kitchen, in your well, in your pipe, you can take the water and you can simply perform. You can check the water, whatever you are drinking is safe to drink or not. That is what I told you. This paper is very important, because it is not studying just for scoring the mark. It is studying to understand the daily health requirement. Okay, that is very important. Good quality water you need to drink. So this is a microbiological analytical procedure which uses the samples of water, and these samples determine the concentration of bacteria in a particular water. What is the concentration of the bacteria that is present in this particular sample? And this procedure is used to confirm that the water is safe for drinking. Okay, the whatever the water you are drinking, that is safe to consume, or that water you are bathing or recreational water are safe to use. Okay, the water you are taking bath, okay, the water you are taking cleaning, the water you are using for washing, all those water should be clean. Okay, so otherwise it will create a lot of issues. And how will you test this? What are the microbiological methods for this water quality testing? That is what we are going to see. This is what this is one of the approaches are the one in which most of the companies they are utilizing. Okay, and it does not require a lot of infrastructure, and it does not require a lot of knowledge. It requires a simple knowledge for testing. Okay, the first test is known as multiple two fermentation test, or most probable number, or MPN, most probable number test. That is known as multiple two fermentation test. Next one is known as membrane filter technique. Next one is known as standard plate count, or aerobic plate count. Okay, so these are the major types of main test that can be used to determine the quality of the water. Now we are going to see one by one. In microbiology, these are big units. Okay, because students will be, if you ask your final year students, your friends, they are going to do major experiment also. Whatever you are studying there, they are going to do experiment. 3 days will take to complete their experiment. Okay, so that is the interesting thing. That is what they are going to do in their laboratory. So first one, multiple two fermentation test, or most probable number. This is one of the oldest method in water, sanitary analysis. Okay, so checking the quality, this is the oldest method. And most probable number is a method used to estimate the concentration of viable microorganism in a sample by means of replicated liquid broth growing in tenfold dilution. I know that you did not understand much. Okay, I will tell you that. To estimate what? The concentration of viable. Viable means a living microorganism. How many microorganism that is a viable or living microorganism is present in the water sample by means of a replicate liquid sample. Okay, whenever we dilute the water, okay, the water sample you dilute it. So even though that is diluted, at what proportion you are going to dilute in tenfold dilution. Okay, in a tenfold dilution, what is the number of bacteria that is commonly living? Okay, a viable living bacteria, that is known as a most probable number method. And this MPN is most commonly applied for the quality testing of water, that is to ensure whether the water is safe or not in terms of bacterial count. Okay, whether the bacteria is safe for human consumption or safe for use. Okay, and I told you what we are going to do. A group of bacteria commonly referred as fecal coliform, which act as the indicator bacteria. Here we are going to detect in order to check the water is safe, we are going to detect the fecal coliform, mainly E. coli. Okay, and that can act as the indicator bacteria. So the presence of very few fecal coliform bacteria would indicate the water probably contain no disease causing microorganism. So I am not telling that the water should not have fecal coliform. There will be fecal coliform. Okay, so none of the water, if you accept sterile water, most of the water you will be having fecal coliform. Okay, whatever the water, the pond water, well water, river water, okay, and the purified water may not have. Okay, so all the water, natural water will be having fecal coliform. But what is the main important thing? It should have a low count that is prescribed by WHO. It should not have a high count. Okay, so then only we can call it as that water is safe. Okay, then the water may not have that much of disease causing organism. Then the presence of, then that is another way. If low fecal coliform, if you detect, there are very less chances of disease causing microorganism. But the presence of fecal coliforms are very, very high, then of course you can expect a very high probability that the water would contain disease causing microorganism, and which make the water unsafe for human consumption or human use. Okay, so the presence of large number of fecal coliform, if you detect by this method, that is clearly indicating that the water is not good for or unable to use for human purpose, or we cannot drink the water. Now, what is the principle of MPN test? This is very, very important. Okay, so whatever the water you wanted to test, you take a sample in a small bottle. You take a sample 100 ml of water you can take in a small bottle from a pond, from a tap, from a well, or okay, wherever you want. Okay, any water you can take. Okay, the water to be tested, diluted serially. You should serially dilute and inoculated into a lactose broth. A broth means this is a medium in which we are cultivate. Liquid medium that is using in microbiology is known as broth. Okay, so that you need to do, just you add, when this addition in microbiology term, you need to call it as inoculation. Okay, so inoculation means you are adding the sample, whatever the water you want, that water you add in a lactose broth. Then that is the interesting thing you can see. If coliforms present, okay, in water, and what will happen? The lactose, that is already lactose is present in the medium and produce acid and gas. You know that contaminated water you drink, immediately you will be having gastric problems. Okay, why it is happening? The bacteria enters into the body, they utilize carbohydrate and they produce acid and gas. So that is why you will be having this lot of dehydration and loose motion and gastric trouble, gastroenteritis. The presence of acid is indicated by the color change of the medium. In this lactose broth or any other medium, we are using a certain color, pH indicator. Okay, the medium, there is a particular pH, and you will be having a particular colored a substance called pH indicator. If acid produces, what will happen? The pH will be changed into acidic level, then the indicator will change the corresponding color. Okay, so then we can see the medium into a particular color. Okay, so and how will you detect the gas? So I told you how to detect acid. Acid change in the pH. What about gas? There is a small, inside a test tube, there is a small tube, a small, very small kutty tube called Durham's tube. Okay, that Durham's tube we will keep in inverted position. Whatever gas is present, the Durham's tube will collect the gas. In the tip of the Durham's tube will collect the gas. This is the way how gas is collected. Now, the number of the total coliform is determined by counting the number of tubes giving the positive reaction. So, both the color change. Positive result mean there should be acid production and gas production. Then only we can call it as a, coliforms are present. Okay, and how many coliforms, how many tubes you are getting, like that you need to check, and comparing the pattern of positive result to a standard table that is prescribed by WHO. Okay, so that I will show you the procedure. So the number of total coliform, that is determining by counting that number of tubes that shows a positive result, that is both acid and gas, and comparing the pattern of positive result, that is the number of tubes showing the growth at each dilution. In each dilution, the tubes that is showing positive result with the standard statistical tubes. Then MPN test is performed by 3 different tests. It requires 3 days to perform. That is what initially I told. This is a 3-day test. First day, the test is known as presumptive test. Second day, the test is known as confirmatory test. Third one, that is known as the test is known as completed test. Presumptive test, confirmed test, and completed test. This is the principle of MPN test. So, if the presumptive test is positive, then only you need to do the confirmed test. If confirmed test is positive, then only you need to do the completed test. Okay. If presumptive test is negative, then within one day you can complete the test. Okay. Now we are going to see one by one what is presumptive test. Okay. So the presumptive test is a primary screening test to sample water for the presence of coliform organism. First day itself, whether you are going to get an indication whether these fecal coliforms are present in the water you are going to drink. Okay, the presumptive test is a screening test to the water sample for the presence of coliform organism. So we are going to use a particular media for testing. Media means the agent you are going to use for the study. Okay, that's the media. Okay, that is known as a broth. One is known as lactose broth, or another medium called the McConkie broth. McConkie, this is very important medium. Okay, culture medium. We have to cultivate, we have to culture where we are going to grow the bacteria in this medium. Okay, and McConkie broth or laurel tryptose broth, that or lactose broth. Laurel sulfate tryptose broth is the first thing. Okay, so it is very simple. So what you have to do is you have to take a set of three sets of 5 tubes. Okay, so 1 2 3 4. Okay, here you can see test tube. Here inside the test tube you can see 10 ml. You have to prepare this lactose broth or McConkie's broth and laurel sulfate tryptose broth. Okay, so here you are having the first test, first 5 tubes with the 10 ml lactose broth. Say for example lactose broth. Next one, here you have to take again lactose broth you have to take, but that is not 10 ml, it is 9 ml. Okay, 9 ml you have to take into all the 5 tubes. Okay, the third one, third set 5 tubes you have to take a 9.9 ml medium. Okay, so in that what you need to do is we need to, here you can see all the mediums are colored. Why it is colored? Because there is a red color you can see. We are added the pH indicator. The pH is pH of all this medium is 7.2 or 7.3. Okay, then you can see a small cootie tube inside. That cootie tube, small tube, you have to keep inverted position. You can see this kept inverted position. This is a small tube and kept inverted position. 1 2, all the tube you can see there is inside another tube. Okay, this is for the collection of gas. Okay, so now the first tube, now this is the first day procedure. Okay, 3 sets of 5 tube. First 5 tube consisting of 10 ml any medium. Second 5 tube 9.9 ml medium. Okay, first second set 9 ml medium. Third set 9.9 ml medium. Now what you have to do is take the water sample from a pond water or a tap water or the water you are going to utilize in your home or toilet water, whatever the water you want. Okay, in a small you can take it. Then in each tube, the first set, here you have added what? Lactose broth 10 ml, isn't it? All 5 tube you have to add. You have already taken 10 ml medium. In these 5 tubes, so you have to add from the water. Take 10 ml water. So take 10 ml water and add in each tube. So total this tube has 20 ml water. So first tube 10 ml. Total quantity is 20. The second case, second tube it's already having 9 ml. Then from this particular same sample, all the 5 tube you add 1 ml here, 1 ml here, 1 ml here, 1 ml here, 1 ml here. Okay, like that 5 to 1 ml. So total 10 ml. Okay, the third tube you have to add, here you can see you have to add 0.1 ml. Here it is already there. How much it is there? 9.9 plus 0.1, 10 ml. Here 20 ml total. Here 10 ml. Here 10 ml. Okay, so now you have added. Now what you have to do is you have to incubate. Incubate means you have to provide a temperature of 37 degrees centigrade and 1 day you have to keep inside a chamber. Okay, that chamber will provide you 37 degrees centigrade adequately. Why 37 degree? We have already discussed most of the bacteria are mesophiles. What are mesophile? They are able to grow at 25 to 45 degrees centigrade. Optimum temperature is 37. So that temperature it will provide. So one full day, 24 hours, you need to keep all the test tube with the medium. After covering it completely, you have to keep it. Now what will happen to the next day? Okay, so what will happen? all the 3 test tube you can see, depending upon the number of coliforms are present this water, it will you can that day night it will start utilizing the medium, and they will start eating lot of ingredients in the medium, and what will happen, they produce they change the pH, they will start degrade the component, and they will produce lot of acids. Coliforms are acid producers, okay, lot of acid. When what will happen if acids are produced in this medium, pH will reduce, okay. I have already told you the pH is 7.2. So that 7.2 will come down to some around 3 or 4, even 2. Then what will happen? It become highly acidic. Then what will happen? In acidic condition, the color of the medium will change. If it is red, it will change it as yellow in color, okay. That is what you can see here, red in color, and here you can see yellow in all the tube yellow in color you can see. Similarly here also, if acid produced, then 2 tubes have converted into yellow, but this can be 2 or what can be 3 or all 5 can be yellow, whatever it is, that depending upon your total this one, okay. And this will be maximum because that is so much 10 ml sample is there. Here 1 ml is there, you will not get all the tube, or sometimes you will be getting all the tube. If all the tube, if you get then water is highly contaminated, okay. Then here you can see nothing has turned because only 0.1 ml is sometimes here also you will be having this particular change, okay. Now you see this is the acid production. Now, if acid is produced, then what about gas? Gas is there, you can see the Durham's tube is kept in an inverted position. The gas, whatever it is produced, it will collect. You can see inside this tube there are white in color, okay, that indicate gas. Sometimes this tube will float over the surface of this particular tube. Instead of the bottom, it will come out, it will come to the top, okay. Then here all the 5 tubes you are having positive. 5 positive means both acid production and gas production. Here 2 positive means both acid production, 2 they are having the acid and gas is also produced, okay. And here 0 positive tubes and 2 positive, here 3 negative 2. Here you can see 5 positive 2. And this particular test is known as a presumptive test. Now what you have to do? Now you have to count from this particular set how many positive tubes are there. Okay, that is 5. Here it is full 5. In the second set, how many positive tubes are there? Only 2. Okay. Third, how many positive tubes are there? That is 0. Okay. So here the number here it is a 5, 2, 0. That number you count in a particular, instead of 5 you can use 3 also, okay. Here you can see instead of 5 2 you can use 3 also. Here 3, here 3, here 3, then it will be 3 2 0, okay. Not need that always should be 5. You can use 3 tube also. So there are 2 types of tube. One is 3 tube fermentation test, next one is 5 tubes test. Here it is 5 tube, but the result is given in the a 5 tube test, okay. Here you can see here this is the tube, this is the standard of WHO, okay. So here 5 of 10 ml, this is 5 of 1 ml, 5 of 0.1 ml. Out of 5, say for example 3 tubes have turned into positive. First one 3 tubes have turned into positive. From here 3 tubes, second one from here 2 tubes, and from here, say for example here it is 1 tube, okay. Then that MPN index is, that is 3 2 1. So that correspondingly the MPN per 100 ml, that is indicating, this is indicating that 17 coliforms are present, okay. So that is the MPN index. MPN index means 17 per 100 ml 17 bacteria. How will you get this? This is a standard table, okay. This is proposed by American Public Health Association by as a part of WHO, okay. So like that, any combination, here it is, you can see sometimes here it will be 2, here it is 1, here it is 0, then it will be having 2 1 0, okay. Then you will be having only 7. This you look the compare from this particular test, you look how many tubes have turned into positive. Accordingly you compare your result in this particular table, so that you will be able to all possibilities. See, all 0 0 0 0 1 0 0 2 0, all possibilities you can see, okay. So that is what is the thing. So here that is known as MPN standard table, and this particular test is known as a ambient test. So after ambient test, what you are going to get? 100 ml of the water. How many coliform are present? Okay, that is what you are going to get. Now you will be able to see 5, 5, 5. Then it will be number will be very, very high. Okay, so here it will be, that is a 95% confidence. The result is not 100% correct. 95% it is correct. This is not that much accurate. And second one is, if this particular test is positive, then second day is a confirmatory test. Then what you have to do is confirmatory test. So you take some microorganism other than coliforms also produce the acid and gas. Okay, sometimes that acid and gas producers may not be coliforms. Okay, to confirm the bacteria are coliform only, you need to perform the confirmatory test. So there is another medium called lactose broth or brilliant green lactose broth, etc. So what you have to do, the positive tube, you take one of the tube from the positive result, both acid and gas, and what you have to do, you take a few sample and add, that is known as inoculate. Okay, so you inoculate, from that particular tube, positive tube from here, take some portion and add that particular sample to the medium that consisting of either brilliant green lactose broth or lactose broth. Then after that same thing, you have to keep it for 37 degrees centigrade and 24 hours you have to wait. Then next day, if you are getting the gas and acid, okay, gas and acid, then you will be able to confirm. Sometimes first 24 hours you will not get as a gas or acid, then you incubate for 24 more hours, 48 hours you need to incubate. Then sometimes first 24 hours you will not be able to see any result, but when you incubate further 24 more hours, one more day if you keep, you will be able to see the acid and gas. Okay, that is known as confirmed test. If that is there, you can see that is coliform only, because in that particular case only coliform will be able to grow in this particular medium. Then third test, if this thing also positive, then we need to confirm that is you need to prove that is coliform. Then what you will do? Sometimes what will happen is, even if you get the confirmatory test positive, that may not be correct result. Okay, then what you have to do is, then you need to prove. How will you prove? Another medium called endo-agar. Okay, if the medium is liquid, that is known as broth, and the medium is solid, that is known as endo-agar or EMB agar, or that is EMB stands for eosin methylene blue agar. Okay, eosin methylene blue agar. Then what you have to do? From the positive tube, from the third, in this particular step, you have to take one tube and take a sample from this tube and add and just to put a streak. Streak means just by using a typical method to add bacteria to this particular medium. Okay, culture, grow the bacteria in this particular medium. Then next day, if you are able to see, then after this particular step, incubate that particular plate for 37 degrees centigrade for 24 more hours. Okay, so then if you are able to see the growth, okay, in eosin methylene blue agar, then you can confirm that is typical coliform. So this particular step you require the first day. If positive, this particular test, the confirmatory test, you have to do second day, and this thing also positive. In the third test, you have to do in the third day eosin methylene blue agar or endo-agar, that is known as completed test. So once the completed test is also positive, that is no doubt that the water is contaminated and that is not good to drink or use. This is known as most probable number test. And there are lot of advantages and disadvantages of this particular test. First, what are the advantages? It's very easy for the interpretation. By simply looking the tube, you can able to tell. Okay, very easy for the interpretation by either direct observation or gas emission, by simply looking the gas. Okay, because if the gas is produced, this tube will be floated over. Okay, it will be at this, it will come out, it will not keep it in the bottom, it will come out. Sometimes it will come out of the, due to the gas production, it will come out of the test tube. Okay, so that is very easy. Then the sample toxins are diluted. So when you are diluting the water sample, that sometimes water will be having lot of toxin due to the heavy metals and all those things, that toxin will be diluted. Then the effective method for analyzing high turbidity sample. Okay, this is the best method for analyzing the high thick sample such as mud, sludge. Okay, not only the thick water, that water consisting of lot of impurities, that is sediments, etc. Okay, and this method can be used to check the coliform count that cannot be detected by any other method. All other methods are not possible. Such a kind of a sample we can use this particular method. Then what are the drawbacks of most probable number? The main thing, it will take a long time to get the result. I told you 3 days is required. Test, once you are giving the test, 3 days. Okay, then the results are not very accurate. Here you can see 95% accuracy only you will be getting, not 100% accurate. Okay, and it require a lot of requirement and utilities. Here how many test tubes and conical flask you require? This is for only one sample. Okay, so here 3, here 3, here 5, here 5, here 5. Okay, then if 10 sample you are having, then you require only for one test you require 15 test tubes. So imagine if you are having 10 different water sample. Okay, then you will be having 150 test tube you require actually. So that is what is the thing. Then it require lot of requirement and utilities. And I told you there is a chances of false positive result. Okay, sometimes what will happen? You will be getting acid and gases that may not be coliform bacteria. There are so many other bacteria also produce acid and gases. Okay, so that is the problem. So because of all those reasons, this is not an excellent method. Next one is membrane filter technique. This is also another important method. This method is known as membrane filtration. It's very simple. You take a membrane. Membrane means that particular nitrocellulose membrane filter paper. That filter paper will be having very narrow pore. When you are passing that water, when you are filtering to that particular membrane, all the bacteria will be trapped on the surface. Only water will be passed through the membrane. Okay, only bacteria will be, all the bacteria will be trapped over the surface and the water will be passed through the membrane. Okay, that is known as membrane filter. Then you take the membrane and keep it in a medium and you will be getting the bacteria. The membrane filtration technique has become the most common and preferred method of evaluating the microbiological characteristics of water. Okay, this membrane filter technique is the very good method for evaluating the count of the water. The water is passed through a membrane filter. That's what I told you. The water, the filter with its trapped bacteria is transferred to a surface of a solid medium or agar medium. If the medium is solid, that is known as agar medium. Okay, in microbiology we call it as agar medium. Okay, a solid medium or to an absorbent pad containing the desired liquid medium. Okay, even you can do an absorbent pad with the liquid medium. Just very simple. Just to take the filter paper, you pour the filter paper. You pour the water through the filter paper and take the filter paper, because all the bacteria that is present in that water, it might have trapped over there. Then you transfer that particular filter paper to a solid medium and incubate, keep it in incubator, give 37 degrees centigrade and 24 hours. One day you wait, next day you can see bacterial colonies. Okay, this is the what is the protocol. This is what is the water sample. This here you can see a conical funnel. Here you can see this is the membrane. Here you have kept a membrane and here you can see a conical flask. Okay, and this is known as a membrane filter. Okay, so here you can see membrane filter have minute pores. So what you can see, the water you are going to fill here and it is going to be filtered. Okay, through this particular membrane it will filter and all the water that is free of pathogen, free of bacteria will be collected here. Okay, and this membrane, you take the membrane out and this particular membrane just you put in this is known as a petri dish, a small plate like plate where you are going to keep your medium or agar medium. So here you will be having an agar medium. What is this agar medium? You are going to prepare lot of nutrients so that when you are keeping the membrane over here, that bacteria will be utilizing the nutrients, and you can see here this is known as the colonies. Okay, next day you can see colonies have grown here. If colonies have seen, you can see that the water is contaminated. This is known as membrane filter technique. There are also a lot of advantages and drawbacks for this particular technique also. So this is a good reproducibility. We can utilize this thing for any type of water. Single step result we will be getting. Membrane filter you require 3 days, but here you will, most probable number MBN you require 3 days, but here only single step you will be getting the result. Filter can be transferred between different media. Okay, we can transfer to any media. Instead of this filter, you wanted any media if you wanted to use, instead of Makongis agar or nutrient agar, any medium, or EMBL agar, use in methylene blue agar, you can use any medium you can use and you can transfer. Large volume of the sample can be processed and increase the assay sensitivity. How much ever water you wanted to pour, you can pour. Only thing is like you should keep on change the conical flask. Okay. The next one, the time savings are considerable. Okay. Here you can see time you can save. Lot of time you can save. Only one single step is very simple. Just simply pour the water through a filter paper. You take the filter paper and incubate it. Next day you will be getting the bacteria if it is present. Filter paper next day, if it is not, bacteria is not there, the water is free of consumption. Then time saving is considerable. Then ability to complete filtration on site. Here we need not transfer to anywhere. Site itself you can complete the process. Then very low capital cost. Okay, so very compared to MBN you require lot of requirement, but here it is only this much requirement is sufficient. Conical flask, here it is a conical bottom funnel. Okay, here you can see another, this one a Petri plate, then membrane. This membrane is costly. Okay, the membrane, okay, that is the main thing. So only this much things are required. Then drawbacks, high turbidity waters we cannot do. Okay, because when you pass through this high turbidity water, what will happen? The membrane will block. Actually, it will not pass through the water. All the debris will be trapping over this surface. Okay, then the water will not go down. Okay, so that is the high turbidity water cannot be utilized. Then high population of background bacteria cause overgrowth. Here it will be, we are pouring, there is not, in the case of here there is a serial dilution. Okay, 10 ml, 1 ml and 0.1 ml. There are a lot of dilutions are there. 10 times dilutions we are making. Okay, but here it is, sample is original. Okay, there is, the growth will be very thick growth you will be getting. Okay, so the high population of background, in a particular plate you will be having 2000 3000 bacterial colony. It will be overgrown. We cannot identify the bacteria properly. Then metals and phenol can absorb the filter and inhibit the growth. Okay, the filter is very sensitive. The metal in the water, if the water has so many metals, heavy metal will be there or phenol will be that can absorb, that will be inhibit the growth of bacteria. Okay, that will be acting as the inhibitory substance. The bacteria will not grow there. So these are the next important method. The next one is known as standard plate count. Okay, so this is another important method. This method is used to estimate the population density of bacteria in a liquid medium by plating a small and dilute portion of the sample and counting the number of bacterial colony. Okay, standard plate count is the one of the simplest method. Okay, we are going to serially dilute the sample, then you are going to count the bacteria. Okay, then use serial dilution to create decreasing concentration of the original sample that are plated, so that the plate will be created with a low number of bacteria that can be counted individual colonies. So you can count individual colony after serial dilution. Okay, then from the number you can calculate the original cell density of the sample. Okay, the original cell density of the sample can be calculated. This is known as aerobic plate count. So this is, we can check the viable bacterial count. So the bacteria, what are the bacteria that is actively present, the growing bacteria, you can check actually. Okay, that is the important. There are two methods. One is known as a pour plate method. Next one is known as spread plate method. The standard plate count is known as serial dilution method. Okay, so one is pour plate method, next one is spread plate method. Okay, so what do you mean by pour plate method? So just as the name indicate, you are pouring. Okay, pouring what? Pour plate method is usually the method of choice for counting number of colony forming bacteria. Okay, which all are the bacteria can produce a colony. Okay, colony means a species of bacteria that is present. Okay, same type of, don't think like a colony means only one bacteria. Colony means more than so many bacteria, but they are in the same species. Okay, that is known as pure culture. Okay, so that is known as colony. Then fixed amount of sample, say for example 1 ml from the water sample is placed in the center of the sterile, that dish, that is known as Petri dish. Petri is the scientist that they have first identified. Okay, Petri dish using a sterile pipette. Then after that, first you have to take a sterile Petri dish, then you take 1 ml of water, then you add that particular water, then you pour the medium, okay, the nutrient agar or Makongis agar or EMBL agar, and you mix it thoroughly. Then you keep it in the incubator, and next day you will be getting growth. You count the colony. Okay, that's all the things. Very, very simple. First, why it is known as pour plate? First, you are in an empty sterile Petri dish. You are going to add 1 ml water sample, whatever you have wanted to check. Then you are adding the medium, that is the growth medium you are going to add. Then you are adding means pouring, hence it is known as pour plate. Okay, so first you are adding the sample, then you pour the medium and you mix it. Then after that, once the mixing is done, the medium, once it will be keeping for some time, it will be solidified. Okay, it will become harder. That medium you keep it in the incubator for 1 full day at 37 degrees centigrade temperature and for 24 hours. The next day you will be having small dot like things, that is colony you can observe, that you count, that is known as a pour plate. So this is what is the method. So this here we can see, here is the dilution. Okay, here you can see this is 9 ml, 9 ml water, 9 ml water, 9 ml water and 9 ml water. Okay, what you have to do, 9 ml water, 9 ml water, 9 ml water, 9 ml water in 5 to 6. Okay, this is known as 1 by, this is the 9 ml, this 9 ml, 9 ml, 9 ml, 9 ml. Okay, so what you have to do, the first 9 ml to take your water sample. Here water sample is the transfer 1 ml. Okay, that means say 9 ml you are adding 1 ml. So the dilution is 1 by 10. Okay, 1:10. Mix it thoroughly. Then now this total volume is 10, isn't it? Then mix it thoroughly from a pipette. From a pipette you add. Mix it thoroughly from this particular tube. Take 1 ml and transfer to the second tube. Okay, then it is 2 times it is diluted, 1 by 100. That means to say 1:100 or 1:100 or 1 by 100. Okay, then take another pipette, correct it, then shake it properly. Take 1 more ml from this tube after adding the sample, then transfer to the third tube. Okay, transfer to the third tube. That is, water is diluted 3 times, threefold, that is 1 by 1000. Okay, so 1 by 1000. Again you mix it thoroughly. Then 1 ml you take it using another sterile pipette. Then you transfer to the next tube, 1 by 10,000, 1 by 10 or 10 raised to minus 4. Okay, then this dilution is 10 raised to minus 4. Then shake it properly. Then take 1 ml. Then what you have to do is, you have to add the last tube, 1 by 1 lakh. Okay, so this is 1 by 10, 1 by 100, 1 by 1000, 1 by 10,000, 1 by 1 lakh. Okay, 1 last tube you have to take it 1 ml out. Then what you have to do, all the tubes are bacteria are distributed. Okay, then from this particular tube take 1 ml as I said. Okay, so what do you do, take from this particular tube, take in an empty Petri plate, take 1 ml. Okay, then you add the medium like this, mix it thoroughly. Okay, mix it thoroughly, then keep it for a overnight incubation. Next day you can see like this. See, so many colored beautiful bacteria. These are highly dangerous pathogen. 1 ml of the sample, this much bacteria are present. Okay, that is what I wanted to tell you. Second tube also same method. Take 1 ml from here in an empty test tube. Okay, add this, then add the medium, mix it thoroughly, then keep it for a solidify, then keep it for incubation. You will be getting, next day you will be getting like this. The third tube, take 1 ml. Okay, pour like this. Okay, empty test tube, just to pour, then add the medium. Okay, thoroughly mix it. Then mixing should be uniform. Then you will be having the surface colonies. You can see like this. In this Petri dish you can see originally, you can see like this. This is another like that, this tube, all the tube. Then what you do, next day you will be able to see this one, 1:10, 1:20, sorry, 1:10, 1:100, 1:10,000, 1:1000, 1:10,000, 1:1 lakh. Count the number of colonies in all the plate and divide by 1, 2, 3, 4, 5. Okay, that is the total number of, that is known as individual bacteria. There is a name called colony forming unit. Okay, so one colony, this is one particular colony. That means to say, don't think like only one bacteria. That colony has a 1,000, 20,000, 30,000 or millions of bacteria. This is one bacteria. There is only one group. But here you can see another colony, another group, another colony, another group. They have a lot of so many colonies. Similar type of species will be growing in a particular colony. This is known as spore plate. Why it is spore plate? First you are going to add the medium, then you are pouring the medium. Then you are pouring the medium. Then you will be getting. Next one is spread plate. This method is the same. Everything is the same. First you are going to pour the medium. Okay. Here first you are going to pour the sample. Okay. Then you are going to pour the medium. Okay, in pour plate. But in spread plate, first you are pouring the medium and make a plate. On that particular plate, in the top, you have to pour the, from here 1 ml you have to pour here. Okay. Then by using an L rod, okay, you can see normal rod you have seen. Okay, L rod, you have to, by touching the L rod, you have to mix it. Here, first you have already prepared the medium. You have made it into solidification you have made. After that, you are going to pour the sample. By using an L rod, you are mixing. Then the process is the same. Next day you can see like this. This is known as a spread plate. Okay. This is what I told you. You have to take a sample from this particular tube. Okay. Then what you have to do? 0.1 ml you have to add. Here it is 1 ml, but spread plate you are always adding 0.1 ml or 100 microliter. Okay. So this is a solid medium, solid agar medium. Okay. The same process. This is what is the same process. From water sample serially you are going to dilute it. Same thing. Okay. Then what you do? You can see an L rod, a small L rod. This is the medium. Once you are pouring that 0.1 ml in the top of the agar, you have to rotate. Okay, you have to rotate like this. Properly using this L rod, spread it completely. Spread it nicely. You need to spread it complete by round. Then you have to keep the plate for incubation. Then you can see the bacterial colonies on the next day. This is original plate, like this you can see. Okay, so that is known as spread plate. So these are the major approaches for by using all this method. What you can see, you will be able to see whether the bacteria, whether the water sample consisting of bacteria or not. Fecal coliforms are present. All those things are fecal coliforms. Okay, that's all, dear students. We can conclude today's class. So today we have discussed indicator microorganism. What are indicators? What are the criteria used for? Criteria can be considered in order to tell that this bacteria is indicator organism. What are coliforms? The total coliform, fecal coliform, etc. How will you test the sanitary examination of water? How will you test drinking water? In your any water you can do like this actually. One is multiple two fermentation test or most probable number test. Then it has the principle. This is a presumptive test, first day test, confirmatory test, second day test, and completed test, third day test. What are the advantages and disadvantages of MPN? Then next one is membrane filter technique, and what are their advantages and disadvantages? Why it is known as membrane filter technique? Third one is standard plate count, okay, or aerobic plate count, okay. That is the thing, okay. Then a pour plate and a spread plate, okay. This is pour plate and the spread plate. So that is what I wanted to tell you. Then these are the textbook, microbial ecology by Atlas and Bartha, or any microbiology book. Okay, so thank you, dear student. Water is so precious. Okay, you can see this kid is drinking the pure water. Okay, and we don't prefer this plastic bottles. Microbiology, when we are talking about this plastic bottle, we are avoid. Okay, water is safe to drink. The water should be safe to drink, and water is the most precious material in this world. Okay, you should preserve water, and by learning these particular things, you should know that how to test the quality of the water. Okay, so that's all, dear students. Thank you very much for watching this particular class. If you are having any clarification, you can ask me. Thank you very much.